My name is Joel Nitta, and I'm presenting on an open and continually updated Fern Tree of Life. You can find the slides online if you're interested at the URL in the title there, and then I just pasted it in the chat. Okay, so building a complete tree of life has been a major goal of biology ever since the time of Darwin. And fortunately, these days, thanks to the extremely rapid growth of data on Genbank, this may become a possibility in a somewhat near future. However, this is, again, because of these very large amounts of data, it's not possible to do this without the help of automated pipelines. And so there's been several automated pipelines that have been developed in recent years, such as these ones, PyFlod, SuperSmart, SuperCrunch, etc., that can take a taxonomic group and then automatically build a tree from sequences on Genbank. However, this sort of approach does not come without some challenges. So one problem is that with an automated pipeline, there's always going to be a trade-off between scalability and accuracy. So any automated pipeline, because of this large amount of data, has to make some shortcuts and assumptions. On the other hand, manual inspection of all sequences would be expected to lead to high quality results, but that sort of approach simply does not scale. Another problem is that a tree built using one of these methods will likely go out of date quite quickly, and that's because of the rapid accumulation of data on Genbank. So we have to keep that in mind when building such a tree. So the approach that we've taken for this study is to try to combine some aspects of both automation and customization. And the goal here is to construct a pipeline to generate a maximally sampled, high taxonomic quality phylogeny of ferns. So why ferns? Well, ferns are a large, diverse, and ecologically important group of plants. They also happen to be my study system, so I'm quite interested in trying to build a maximally sampled fern tree. And if we were to compare them to some similar organisms, such as seed plants or angiosperms, ferns are actually just easier to work with because of their overall lower species number. So ferns include about 12,000 species, of which somewhere around 40 to 50% have been sequenced. In comparison, seed plants include over 300,000 species, so order of magnitude more, and less than 20% have been sequenced. So ferns are just more tractable to work on. And that's not to say that other people aren't trying to build completely sampled seed plant tree of life, which is really important. But here we're focusing on ferns. So one thing that I want to mention before moving on to the methods is that Sanger sequenced plastid genes have really been the workhorse of fern molecular systematics for the past 20 to 30 years. So this chart shows the number of sequences in Genbank, number of fern species in Genbank since 1990. And of course, we see that it's steadily increasing. And you'll also notice that I've split this up by genomic compartment. So plastid, nuclear, and mitochondrial. And that plastid sequences account for, by far, the vast majority, in fact, almost the total of all species in Genbank. So if we're trying to build a maximally sampled fern tree, plastid sequences are going to be the data source of choice. Okay, so this is actually an overview of the complete workflow. And I'm going to go in some of the more pertinent parts of it in more detail in the upcoming slides. But here I just like to stress that this is all automated via code using the targets R package so that we can repeat this regularly as needed. And that the whole thing takes about 10 to 14 days to run. Most of that is taken up by maximum likelihood analysis. And you'll see that there are some points in include this hand symbol, and those are to show steps that include some manual inspection to help ensure that we have a high quality phylogenetic tree. Okay, so I do want to go through the workflow and just highlight some of the uh, parts that I think are more interesting and possibly could be helpful to others. So I'm not going to read these all on this slide just now. I'll go through them one at a time. Okay, so our first step is we download data from Genbank, and here we try to increase the efficiency and reproducibility of using the Genbank data by actually using a local database. And that's done with the REST as R package. Quick aside, I'm now the maintainer for that R package, so if anyone's interested in using it, feel free to ask some questions. So what we do here is instead of querying Genbank Live via their online API, we use the Genbank FTP server and download a complete chunk of Genbank, the plants division. And so that's at a fixed version, and it's not going to change from day to day. So that helps increase reproducibility. We then slim that down to just the sequences that we're interested in, ferns, and save that in a local database. And then when we're querying that database, it's much more efficient to do it locally instead of repeatedly querying Genbank Live. And of course, we post this on Figshare so that others can use it as well, who are also working on ferns. Another feature is that we extract sequences from the Genbank accessions without relying on annotations. And that's because there is no standard for how to annotate genes in Genbank. So the same gene can go by different names or formats, and in some cases, if it might not be annotated at all. So to do that, we use a software package called SuperCrunch, and it involves two steps. The first step is to curate a set of reference sequences. We do one sequence per genus, and we do that by actually using well-annotated accessions from Genbank. Next, we use that as a reference blast database, and then blast all the possible accessions to it, and only keep the portions that match. So in that way, we don't have to rely on Genbank formatting of gene names. Another feature to improve the taxonomic quality of our tree is we implement a custom fern taxonomy. And that's because species names on Genbank were, of course, not curated with ferns in mind, and they include many many synonyms in ferns. So for the same species may actually appear under different names and for different accessions. And the way we do that is log of life, which has a good sampling of ferns taxonomic names, including many synonyms. But however, we couldn't use it in its exact form. We needed to make some updates and changes so that it could be suitable for our project. So we started from those and then created a new taxonomic database called Teridicat, which is specifically designed for ferns and is consistent with our tree. And here I just want to mention another R package that I have developed that may be useful to others called DWC Taxon, and that's used to curate data that are in compliance with the Darwin Core taxon standards. The next step after curating our reference database is then to resolve those species names in Genbank to that. And there I also have created another tool called Taxa that allows us to resolve names to a custom database. So many existing name resolution tools for species names do not allow you to do that. You have to use an existing database. So this one is more flexible. Okay, so that is how we generated our reliable set of fern species names. So after these, after assembling these data and cleaning up species names, we finally get to do the phylogenetic analysis. And that's done in two steps. The first step is using plastome data. So this includes 79 plastid loci, so from the chloroplast, and about 400 species. And we first do a maximum likelihood on that and on that data set to assemble what I call the backbone tree. So these plastome sequences are span the deep divergences in the tree, and by doing that, we can obtain a really robustly resolved backbone phylogeny. And then the next step is to use that as a constraint in analysis of the Sanger data set. And the Sanger data set includes many more species, 5,000 species, but just 7 loci. So that then what the Sanger data set does is help to fill in and get maximal sampling along the tips. So by this approach, we can have robustly a robustly resolved tree along the backbone, and then maximal sampling along the tips. And I would just mention here that originally we also had tried a supermatrix approach where we had a combination of the plastome and Sanger sequences together in a single really big, really sparse matrix, but that just took far longer. So this two-step approach maximizes accuracy in sampling and is also a lot faster. Okay, throughout the pipeline, I also want to mention some checks that we incorporated to try to eliminate what I call rogue taxa, and those are typically misidentified species on Genbank. So an automated approach that we took was to do an all-by-all blast. So for this, we first constructed a blast database of all possible sequences, and then we query each sequence in turn against that. And any sequence that matched to the wrong family, we flag and remove as a misidentification or a rogue. And in that process, we found about 70 accessions that seem to be clearly misidentified. Another check that we did in addition to this automated check is a more manual check. And prior to doing the final phylogenetic analysis in IQ-tree, we ran IQ-tree in fast mode, and that finishes in just a few hours as opposed to like 2 weeks. And so that gives us a chance to inspect the initial tree and look for any more rogue taxa that may have fallen through the cracks and not been detected by that automated step. Because the automated step only works at very rough levels, right, at the family level, whereas in order to find other rogue taxa, we actually do have to look at the tree. All right, so those were some features of the pipeline, and now I'd like to show our results. So this is the Fern Tree of Life, or FTOL, version 1.1.0. It includes 5,582 species out of an estimated 12,300 or so extant species, so getting close to 50% sampling at the species level. And this is a 40% increase relative to the previous largest fern phylogeny. And important thing to stress here is that this is an increase, which is great, but we don't want to have this one tree be finished and not do anything more with it. We do plan to continuously update this. This is to show that we do have a well-resolved tree, robustly resolved tree. You can see that we have high bootstrap values in yellow across the tree, and particularly at the deeper nodes. So one interesting result is that we conducted a divergence timing analysis using penalized likelihood with TreePL. And we did that using an expanded set of fossil calibration points. So we had 51 fossils, which is about 2 times more than any previous study. And as a result, we were able to push back stem ages for most fern families from around 10 to 30 million years. And for a long time, since some of the first molecular dating studies in ferns, it had been thought that the majority of extant fern lineages diversified around the same time as angiosperms and probably in angiosperm-dominated forests. But our results actually may contradict that, or at least do not support it. So we need to look into this in more detail, but these are some interesting preliminary findings. And this is a graph showing older stem ages for most families you can see we have all of the families lined up on the y-axis there, and our ages, which are the red points, are older than the other, for most families, than the other previous studies that we're comparing. So one really important aspect of our study is that we want to make our tree a tool for other researchers. And so we've provided it on a web portal at fernfy.github.io. So if you go here, you can see we have a downloads tab where you can download the data in zip form. And we also have a data explorer. So this is a Shiny app built with R that is there to help people who want to find more information about a particular species. So if they use the tree and they notice a species maybe seems like it's in an odd place and they want to know where the data came from, you can click on here, select a species or type in its name, and it will tell you the accession and information about the taxonomy and how sequences were joined, that sort of thing. And we also want to make this, add some more functionality going forward to make it even more useful. We've also made our tree and the associated alignments available via an R package called FTOL R. So this makes it really easy to load the tree directly into R, and it has some additional customizations, like you can drop the outgroup, choose specific loci from the alignment, etc. And another important feature of this project is that we want to get involvement from the botanical community. And one example there is that we consulted with an expert on one family, Philipteridaceae, that had a lot of non-monophyletic genera in an earlier version. And by doing that, we were able to subsequently decrease the number of non-monophyletic genera from 16 to 7. So we want to continue to get input from improve this and make it a more useful tool for everybody going forward. So in summary, I think that FTOL hits a sweet spot between automation and customization, at least for trees at roughly this scale. So we have automated version mining of Genbank data, but then we combine that with custom taxonomy for ferns and input from taxonomic experts to generate a high quality tree that's immediately usable for people working on this group. So in the future, I have several goals. First of all, I do want to complete the full tree of life and fill in that missing 50% of species. To do that, I think we can't just wait for sequences to show up on Genbank. We'll probably need to do some additional sequencing, perhaps of herbarium specimens via plasmon skimming. I want to integrate the FTOL with the pteridophyte phylogeny group 2 taxonomic system, which is a taxonomic system at higher levels, genus and higher, so that that can be also an online and living document that will always reflect the most up-to-date hypotheses of fern phylogeny. We want to transition to phylogenomics for all species instead of just using Sanger data. And then I also want to continue to provide tools such as R packages that will contribute towards building the full tree of life. So with that, I would like to thank all of the institutions and individuals who made this possible, and you all for listening to my talk. Thank you very much.
And I think we may get cut off soon, but we have some questions.
Hi, Joel. Very nice talk. Thank you. I have a question about the strategy with the constraint. And did you try to run a version just without constraint, also using the fast mode to see what sort of like difference in likelihood do you get, and how much, how much that constraint impacts the likelihood?
Okay, yeah, thank you. I have not tried that, but that's a great suggestion. I did, like I said, tried it on a super matrix including all the plastome and the Sager sequences, and we obtain similar results, but it takes a lot longer.
How about constraining just to the few plastic like universal markers on a full matrix?
So I'm not quite sure.
Well, I understand that you have a subset of the data which has many more marker genes, and then yeah. So how about if you just constrain yourself to like the plastic gene that are also found in most of your species, do you get a substantially different tree?
You mean for the plastome, like for the plastome sequences, if we only restrict it to the 7 loci instead of the full set?
Right.
Is that the question?
Right.
Yeah, honestly, I'm not sure. I'd have to try to find out. Yeah.
Okay, thank you.
But like I said, those markers are the ones, of course, that have been used for fern systematics the most over the past 2 decades. And so I wouldn't expect things to change a whole lot, is my gut feeling, but no way to know unless I try.
Thank you.
And I'm not sure if we're cut off from recording, but I have a quick question if you don't mind, which
Please.
is about the sort of rogue taxa, where you said there were, I can't remember, 40 or so taxa that seem to be in incorrect families. You have like fields for voucher information inside your Shiny app, and I'm curious about what proportion have voucher, and especially whether the bad actors, the rogues, had voucher information where somebody could go look at them and say, yeah, this is screwy.
Yeah, thank you. That's a great question. I did not try to actually look at that and track down those further than that. I will look into that. I would say though that unfortunately, most accessions in Genbank, I'm not going to put a number on it, but certainly the majority, well over a majority, do not have voucher information with them. So so of course this tree has 1 tip per species, and sometimes you have multiple accessions for a given species in Genbank for different genes. So how do we join those together? Well, instead of just say like taking the longest gene, the longest accession and putting them together or something, we could have misidentifications. We could have, there's all things that can go wrong. So I tried to be more careful about that. And for example, I use that voucher information as one criterion. So if we have multiple accessions that are all from the same voucher, then I can be confident in putting those together. So I do use voucher information when possible.
And I think that's a really good idea. And my experience in looking in other groups is, yeah, people ignore putting voucher info in, and well, they shouldn't. So
yeah, there's some fields that I wish were official Genbank fields, but they weren't there to begin with, and that's what we have now. So